Hello. I'm just leaving the last lecture. Oh. Don't worry, I'm not taking over. Who's coming to lecture, by the way? Oh, okay. I'm just concerned because there's a hole in the floor that I kept falling into, which wasn't. It's very disconcerting. Yeah. It's very close to where he stands. All of a sudden, hey, so there's a hole in the floor. You might want to be aware of.
Beg your pardon? Oh, yeah, I know. It's a little iffy, isn't it? Yeah. Yeah, I almost fell down it yesterday. So, yeah.
Oh, well.
Well, good morning, everyone. Nice to see you again. I think this is my final lecture in this unit, so I've probably left the toughest topic till last. And perhaps I should have got Alison Imrie to give this lecture, because it really does need an immunologist rather than a sort of toxicology person to teach this topic well. But I'll try to do the best I can, and perhaps those of you that do have a strong immunology background will understand this topic better than I can, but I'll just go through what I consider the basic aspects of this phenomenon. So what we're talking about today is a very serious problem clinically, and it's serious because we're not very good at avoiding these problems and predicting people that are at risk of them. So it's this issue of an adverse drug reaction. And an adverse drug reaction, defined by different bodies as essentially unwanted harmful effects of drugs that occur at doses normally used for treatment. That's the important distinctive feature of an ADR. So the Australian Medicines Handbook, the Bible of drug use, particularly if you're a pharmacist in Australia, defines an adverse effect as a noxious and or undesirable effect associated with the use of a drug at doses normally used. So this is the essential idea here. And of course, bearing in mind that it's not always easy to say what exactly is the normal dose of a drug that's used in a patient, because in a population of people, doctors will find they have to administer different doses just to get a clinical response in different individual patients. So defining what's normal isn't always easy, but the basic idea is that a adverse drug reaction is distinguished from toxicity. Toxicity is usually unwanted effects that occur at what we might call supratherapeutic. So supra means above or beyond, so above the therapeutic window, associated with the use of higher than normal doses, whereas an ADR is an unwanted drug effect that can occur at normal therapeutic doses. Now these are complex, and we'll look at some of the complexity in the rest of this lecture, but the basic idea and why I'm including this lecture in a pharmacogenomics series is that the hope is, and it certainly works for some drugs, that by doing pharmacogenomic testing, particularly of HLA alleles involved in particular exaggerated drug responses, we can minimise the risk of these ADRs and avoid problems. Okay, you've probably heard about ADRs in other lectures. I think it's probably covered in second year pharmacology. But the basic idea with an ADR is that they tend to be classified as either type A or type B. Type A are pretty predictable, and they're usually just exaggerated pharmacological responses to the primary mode of action of the drug. So some classic type A ADRs are things like muscle weakness in people with cholinesterase inhibitors, entirely predictable. Heart block in the case of people on beta blockers like propranolol, hypoglycemia in recipients of insulin and so on. So the basic idea here is that for some reason or another a person is showing exaggerated sensitivity to the predictable side effects of the drug. Okay, so thankfully that's about 85% or so of 90% of ADRs of that nature, and they can usually be managed just by dropping the dose or switching to another pharmaceutical, and there's no long-term consequences for the patient. But much more serious are the type B ADRs, and the B stands for bizarre in contrast to A for augmented. And the basic idea here is that a bizarre drug reaction is one that's not necessarily related to the dose administered, so you can get an ADR, a type B ADR at a very low dose of a drug. And they're idiosyncratic in the sense that they reflect something unusual about the patient rather than about the drug. So they're not related to the primary pharmacological effect of the drug. Something else is happening. And also there's a strong role for immunological mechanisms in the average type B ADR, but not all of them. Okay, so this is probably one of the major challenges in pharmacology, this problem of how do we minimise the prevalence of type B ADRs and manage them and hopefully eliminate them. And so this is a significant problem, counts for some 100,000 or more deaths globally every year. So it's a significant problem in modern pharmacology. We often say that they are unpredictable, and that I guess you have to understand the context in which that occurs, but there are some type B ADRs that probably can be predicted on the basis of genetic testing, and so that's what we're trying to consider in this lecture, but be aware that that doesn't work for every type of ADR, type B ADR. Now, another important feature here is that ADRs, type B ADRs often involve the skin, and that's because the skin is so loaded up with immune cells, and it's a frequent participant in drug-induced toxicity. So around 50% of type B ADRs involve dermatological manifestations. Thankfully, a lot of those are fairly rare, so you don't need to panic every time you get a rash to a drug, because most of the time they're mild and they will disappear with cessation of drug or switching to another drug. So a classic example of this condition is just a generalised sort of reddening, and this maculopapular exanthemas, or MPEs, and these are the most common hypersensitivity reactions. And while they may incur immunological mechanisms, they're not necessarily, it's really just a non-life-threatening allergy, nothing to worry about. Now, the more serious conditions, though, you should worry about. You don't want to have one of these. So these sometimes called severe cutaneous adverse reactions or SCARs, and some very nasty conditions indeed. So we'll look at these in a minute. AGEP, DRESS, SJS/10. So these are often a spectrum of conditions that are lumped into different categories. Now these are the ones, although they're very serious and potentially life-threatening in some cases, they're also where we hope we might achieve reductions in their prevalence with the use of preventative pharmacogenomics screening. Okay, so let's classify ADRs then. We're particularly worried about these bizarre type B ADRs that account for perhaps 10 to 15, maybe 20% of adverse drug reactions. They're often hypersensitivity reactions, and they can either involve non-immunological mechanisms, non-allergic, or immune-mediated, and then we attach a name allergic to that sort of ADR. Now, metabolic abnormalities can be some we've already talked about, really. P450 deficiencies or transported, variant transported genes, such as we've looked at in the last couple of lectures. They basically fit into this sort of, or they can give rise to clinical conditions and clinical responses to drugs that fit into these sorts of non-immune mediated hypersensitivity reactions. And then there are also inherited genetic conditions that involve or that sort of limit the ability of the body to respond physiologically to the presence of a drug. A drug puts stress on physiological processes in the body, and sometimes people with inherited conditions may show an adverse effect simply because they're lacking in some metabolic pathway. So some of the porphyrias, inherited variant forms of involved in porphyrin production. There's a whole bunch of those, and so they are an example of these sorts of adverse reactions that can occur with drugs in people with genetic inherited traits. But really what we're interested in this lecture are the immunologically mediated adverse drug reactions, the true drug allergies, and in particular we're going to focus on those that aren't necessarily immediate in onset, and these are the ones where we get benefit by HLA pharmacogenomic testing, at least we hope we can. Okay, so here's a nice little summary of the factors that contribute to drug hypersensitivity reactions, and like I said, some of this we've considered in our previous lectures in pharmacogenomics. Inherited variant forms of drug metabolizing enzymes or inherited variant forms of drug transporters lead to some individuals having higher than normal concentrations of drugs in the blood, and this can be a factor in predisposing people to some of these nasty severe cutaneous adverse reactions, but also DILI. I think I talked about DILI, drug-induced liver injury, a while back, maybe in first semester, where the liver is the target. So the skin and the liver are the two most common targets for hypersensitivity reactions. But what we're talking about particularly today are these T-cell mediated toxic phenomena that involve activation of T-cell mediated destruction, and this is particularly problematic in the skin because they often involve priming of the T-cell system and migration of memory cells to skin, and then on re-exposure to the drug they can mount these very, very nasty cutaneous reactions. Okay, so the thinking is that these drugs in these susceptible individuals, and usually it's because they've got inherited variant forms of some of these HLA family proteins, usually class 1 family protein variants, and these class 1 proteins, HLA proteins, are involved in presenting peptides by antigen-presenting cells, and this is often a keratinocyte in the skin that does the job of an APC, and for whatever reason it presents a peptide to a T cell receptor on a T cell, and this activates this downstream inflammatory and immune-mediated process that can be very serious in some cases because it triggers apoptosis and necrosis of keratinocytes in the skin. Okay, so we've identified some of the variant genes at the HLA locus, we'll come to that in a minute, that predispose people to some of these nasty toxicities. Now remember I said back here in this diagram that we are distinguishing between non-immediate and immediate reactions. And these immediate reactions can also be quite serious sometimes. Adverse drug reactions that involve, say, anaphylaxis or urticaria, that is swelling, the formation of these skin lesions that are fluid-filled, bronchospasm. These are often very rapid in onset, and they're triggered within less than an hour of ingesting the drug. And they can be very serious and life-threatening because if you get bronchospasm, it can the patient can literally choke to death. So it's a medical emergency. But if you survive that, if you get appropriate emergency medicine intervention, then you can often recover quite quickly from those sorts of conditions. Okay, so we're not looking at those IgE-mediated phenomena today too much, but what we are focusing on are these scars, these severe cutaneous adverse reactions, some of which can be quite nasty indeed. So there's this spectrum of conditions on a sort of a continuum. Stevens-Johnson syndrome is the more mild end of the spectrum. Toxic epidermal necrolysis is up the other severe end of the spectrum, and we'll come back to that in a moment. And then there are people that fall in between both of those sort of extremes. Okay, so these are more nasty and sometimes can have mortality of over 50% in some severe cases of 10. Okay, so I can't claim to be an expert in this, and you really do need a good background in immunology and dermatology really to teach this topic properly, and I freely admit I don't have that, but it seems to me that one of the issues in thinking about drug-induced allergy is how long does it take for the syndrome to manifest itself upon challenge by a drug to a patient that's undergone priming of their immune system. And if it's an Ig-mediated cell response, particularly involving mast cells, it often shows up within less than 6 hours or perhaps even just 1 hour. So the timing of the onset provides immunologists with a bit of a clue as to the clinical condition we're dealing with and the predominant immunological mechanisms involved. So some of these other nasty conditions, including drug-induced liver injury, but all these other skin conditions such as AGIP, DRESS, drug reaction with eosinophilia, systemic symptoms, Stevens-Johnson syndrome, 10. There's often a delay of days or even weeks before these conditions manifest themselves. And there's also some of these conditions differ in terms of their prolongation, the duration of adverse manifestations. So some of them last for weeks, months, even years perhaps, but then others, such as those IgE-mediated conditions that you can recover quite quickly with appropriate clinical intervention. Okay, so for the rest of the lecture I'll just focus on these nasty, severe skin reactions, scars, severe cutaneous adverse reactions, all of which involve T cell activation and aberrant presentation of antigens to T cell receptors. But the thing that differs between these three broad classes of scars is the nature of the T effector, the nature of the dominant T cell involved in the clinical syndrome. And this is where my immunology starts to break down. And but you can see at the top of this list are relatively survivable conditions such as AGEP, acute generalised exanthematous pustulosis, bit of a mouthful. But these are basically sort of generalised reddening of the skin with little pustules, sterile pustules. So a microbiological origin can be ruled out. They're sterile, but it's just inflammation with predominantly neutrophil involvement as well as mild eosinophilia as well. So the dominant T helper cells here come from the Th17 family, and the dominant effector molecules, cytokines, include a combination of factors, cytokines, GM, CSF, IL-8, IL-17, IL-22, interferon gamma, which plays a role in all of these conditions, and then TNF-alpha. But the important thing from a clinical point of view is that these AGEP type syndromes are relatively survivable, quite survivable. Mortality rate of less than 9, or greater than 90%, or less than 1%. Now the more serious conditions are the DRESS and DI, drug-induced hypersensitivity reaction spectrum, and these are Th1 and Th2 dominated pathways, and so now we're starting to get the involvement of things like IL-5 and IL-13, and also strong clear roles for TNF-alpha. These have a higher rate of mortality, more serious conditions, but there's extensive rash over greater than 50% of the body surface area, but also some systemic effects, but usually not as mild and not as severe as we see in some of these quite nasty conditions down the bottom here, such as Stevens-Johnson syndrome and toxic epidermal necrolysis. So these are really life-threatening because they're dominated by CD8 cytotoxic T cells, which are, as far as I can tell, the bad guys of the T cell repertoire, and they activate extensive apoptosis of keratinocytes and necrosis of keratinocytes with some pretty nasty pathways here. We'll come back to in a minute. Granzyme B, perforin, and granulysin. That'll probably mean more to an immunology person than the rest of us, but these are nasty pro-apoptotic pathways that just kill off massive numbers of skin cells. And the poor old patient, if it's a severe case of 10, they can, toxic epidermal necrolysis, they can lose in excess of 30%, even 50% of their body surface area, just completely shedded off. And this is shockingly life-threatening because the skin plays a very important part in keeping body constituents in the body, so they're basically very vulnerable to water loss, fluid loss of a life-threatening nature. Okay, so it's a full-on medical emergency. Okay, so here's a nice little summary of these three broad classes of T-cell mediated toxicities, rare toxicities involving skin reactions. And I recommend, if you're interested in immunology, having a look at this recent review by Gibson and Associates with a strong West Australian involvement. There's a group down at Murdoch, Elizabeth Phillips. She's a Canadian, but she's one of the leaders in this field internationally. She's got a joint appointment in pharmacology at Vanderbilt University in the US, and she does some very nice work in this area. So anything by Dr. Phillips is really, Professor Phillips is well worth reading, because she really is moving this field along from a clinical classification point of view in terms of understanding the immunological basis. So basically, in a one of these milder AGEP type conditions where we see a neutrophil dominated environment, and IL-8 is one of the key mediators involved in activating neutrophils and promoting their recruitment, and as well as the TH17 helper cells are involved as well. You can see the condition here, so it's this generalised reddening of the skin, and then these little white elevations are known as pustules. And like I said, most of the time, if you stop using the drug, this condition will reverse, and the patient will make a good recovery. Coming to DRESS and drug-induced hypersensitivity, we get a little more serious because we're now getting extensive involvement of memory cells, T cells in the skin. There's a process of priming involved, and fundamentally this activation of T cells, but it results in this sort of a mixed Th1, Th2 type phenotype. It's associated with chronic inflammation. But the really nasty ones are these toxic epidermal necrolysis and Stevens-Johnson syndrome. And you can see some of the nasty signs here. There's often extensive damage to mucosal areas, the mouth, the anus, the respiratory tract. Really is a nasty condition. And it's nasty because it's dominated by the cytotoxics, CD8 cells, and they trigger this process of by recruiting natural killer cells, activation of these cell death pathways involving these nasty mediators of keratinocyte death. So extensive blistering of the skin and extensive shedding of the epidermis leaves patients very vulnerable. Okay, so because of the severity of some of these cutaneous reactions to drugs, obviously there's been a lot of effort by pharmacogenomics experts trying to work out why is it that such a low proportion of people are vulnerable to these very nasty skin manifestations when millions of other people use those drugs every day, you know, without any adverse effects. What's going on to cause these unusual responses? So people have identified a number of variant genes that seem to confer an increased risk of some of these conditions, and we don't have time to go through all of this, but like I mentioned earlier in the lecture, there's a role for some drugs for altered gene variants for P450 genes, and glutathione S transferases can also predispose some people to these sorts of dermatological reactions, transport of variant genes. And then in the antigen presenting cell, typically a keratinocyte for most of these conditions, variant genes in variants within the proteasome constituent genes involved in proteolytically degrading antigens, and then to form the peptides. So that some people can have altered processing, intracellular processing of antigens. But really the most significant ones that have the clearest and strongest association are basically HLA1 class 1 variants, and these are the ones that seem to confer altered T cell activation on people in response to use of some of these medications, and we'll look at some of those mechanisms in a moment. And then there are also, as the CD8 cells, cytotoxic cells get activated, some of these other participants in immunodestruction. People have also found variant genes in some of these mediators of apoptosis that alter a person's susceptibility to drugs too. But most of the time, as far as I can tell, the ones that are really strong in terms of their statistical association with the clinical condition involve HLA1, class 1 genotypic variation. Okay, so what are the drugs that are involved in most cases of the severe epidermal necrolysis type responses? Well, it thankfully doesn't occur with all drugs, but there are particular families of pharmaceuticals that are notorious for causing these conditions. So various antibacterials, penicillins, particularly notorious, sulfonamides, don't use them so much anymore, but penicillins of course remain in widespread use. Anticonvulsants are also a significant class of drugs associated with this condition, things like phenytoin, carbamazepine, long-standing drugs in the use of in the treatment of epileptic seizures. Various NSAIDs such as diclofenac, and then antiretrovirals, including abacavir. And the first association of a HLA variant genotype with abacavir hypersensitivity was actually done here in Perth at Royal Perth Hospital back in about the mid-1980s, mid-1990s I think it was, and it sort of set a lot of this field alight. So it helped increase attention on genetic variation within these HLA locus. Allopurinol, the gout drug, is another significant player. Now, so how prevalent are these conditions? Well, this review just came out very recently in a medical journal of Australia, just looking at the number of reported cases of these severe drug hypersensitivity reactions. So we're talking about DRESS here and SJS/10. And for 4 drugs, and you can see the numbers have increased in the case of allopurinol, probably due to just increased prescribing for gout. Gout remains a significant problem in Australia, but gone from 8 cases reported about 20 years ago to 35 in the most recent reporting period. So a total of 93 cases in 20 years. Carbamazepine, about the same, 112 cases. Phenytoin, 60 cases. Lamotrigine, 75 cases. So these are significant numbers of people across Australia experiencing these very nasty dermatological reactions to skin, to drugs. Okay, so those numbers there are for the numbers of prescriptions over the same period. So they're very low incidence. So 93 cases of allopurinol adverse events out of 16 million scripts. So, and overall for all these drugs, 340 out of 20, close to 30 million scripts. So very low incidence conditions. This is what makes detecting these very challenging, because you know, doing genotyping on everyone that gets the drugs is fairly costly and demanding. Takes a lot of resources, so this is one of the challenges in this area of pharmacogenomic utilisation, where predicting exactly why a person might have one of these conditions can be challenging. Okay, so if we look at the overall mechanisms involved in these necrolysis, epidermal necrolysis conditions, essentially there's mechanisms occurring potentially in the liver, but predominantly within the skin, and for whatever reason or other we're getting this T cell receptor interaction with a HLA allele that for some reason is going haywire. And this is, there are several mechanisms, we're going to look at this in a moment, that are proposed to explain why this process goes haywire in certain people. But the basic idea is we're presenting these self-peptides, and this is activating these CD8 memory cells, and that this drives this resulting immune response to keratinocytes, self-structures, including some of these very nasty drivers of apoptotic and necrotic cell death. So extensive keratinocyte death, as well as massive fluid loss due to systemic effects, systemic inflammation. Now the interesting thing is that there's also this so-called heterologous immunity model, and this is an interesting phenomenon. where people that have had a certain viral infection, perhaps in childhood, seem to be more susceptible to a drug-induced severe cutaneous adverse reaction. And again, this strains the limits of my immunology, but it does seem that there's an element of cross-reactivity in terms of the immunological response to the virus and that of the response to the drug. Now, for the rest of the lecture, then let's just talk about these all-important HLA variants. So, this is of course, this highly variable sequence within the human genome known as the major histocompatibility complex. And in humans, we call this the HLA, human leukocyte antigens, basically because this was first described in leukocytes as a determinant of people's reactions with allografts in organ transplantation context. But it's a highly polymorphic region that encodes various cell surface proteins, and these are classified, two of the most important categories in a pharmacology context are class 1 and class 2. And basically these are distinguished by, in the case of a class 1 protein, it contains this transmembrane binding region, and then this alpha 1 and alpha 2 structure that contains a peptide binding groove. And this projects extracellularly from the antigen presenting cell. Now in the case of a class 1 HLA protein, there's this essential role for this participating small protein, an extracellular beta-2 microglobulin protein. This is non-covalently associated with the so-called heavy chain of the protein class 1 structure. But then in class 2 surface receptors, these are basically heterodimers of structurally sort of similar proteins, but they also have this peptide binding group. For the purists, there's a bit of difference in the sort of binding preferences of the peptide binding groove. The class 1 molecules tend to favour relatively small peptides presented by digestion of processed antigens, and the class 2 molecules have a bit more of a larger preference for 12 amino acid peptides or larger. So essentially, now within these various HLA proteins, there are a number of distinct proteins, and you can see them here within the class 1, that are particularly important, HLA proteins that are particularly important as sources of variability as predictors of drug-induced toxicity. So you can see here HLA-B. This is associated with a number of clinical conditions that are unrelated to drug treatment, but down the bottom are the ones of pharmacological interest. So the HLA-B class of proteins, variant forms of those are associated with abacavir hypersensitivity, carbamazepine, SJS10. But then you'll also see over here there's another class 1 family, HLA-A proteins, that are associated with other types of dermatological reactions to carbamazepine. So this is what makes this field kind of challenging. The same drug doesn't necessarily just work by a single HLA variant, a family. Class 2 HLA proteins also have a number of different loci that are, some of which are associated with drug-related conditions, adverse responses, but I don't think the statistical association with class 2 proteins with their relevant clinical condition, a drug-related clinical condition, is quite as strong as it is for class 1 protein. That's as far as I can tell for as a bit of a novice in this area. Okay, so what's going on? Why is it that people with certain variant forms of these HLA proteins particularly class 1 HLA antigens, are so vulnerable to drug-induced dermatological reactions? Well, it's all got to do with the presentation of peptides to the T cell receptor by the antigen presenting cell, and this is usually a keratinocyte in the skin. It's been primed to produce this particular immunological response. And if you look at the top of the groove in a class 1 protein with that peptide binding region, this sort of mechanistic stuff's hard to get your head around, but basically the idea is that if you've got an inherited variant form of the protein, you'll have a different amino acid at one of these sites, and it basically means that you're going to bind a peptide a little bit differently to other people, and for some reason this altered binding of the peptide results in an altered T cell activation response. So T cell responses are very important to normal immunological health, are very important in damping down reactions to self proteins, and also responding appropriately to non-self threats. But what happens in these sorts of cases, people with these variant forms, and they're called risk HLA alleles, risk alleles, are basically prone to sort of inappropriate activation of our T cell receptor because of some altered binding property. Now, so that's one of the theories, but essentially there's been a bit of work over quite a few years now trying to work out what's going on with drugs. Why is it certain drugs activate these sort of abnormal HLA-mediated T cell activation responses and others don't? Well, one of the classic theories is the so-called haptenization hypothesis, and this is pretty familiar probably to a pharmacology person, particularly if you've suffered under my lectures in toxicology, because these often involve formation of reactive metabolites by P450, and these often involve formation of sort of what we call haptens, which is a drug stuck on a protein, so an adducted protein basically. So when that damaged protein gets proteolytically degraded to form the peptides, the presence of the adduct on the peptide seems to have the effect of activating the T cell receptor, because it's presented in a different manner to an unmodified protein. So it activates the T cell via this sort of pathway. So this is one theory, that people with variant or risk HLA alleles basically, for some reason, activate T cells more because in the presence of this adduct on the protein. So have an exaggerated response due to a variation in the presentation of that peptide to the T cell receptor. So this theory works pretty well for many drugs, including beta-lactams like penicillin. So they're some of the most commonly implicated in drug hypersensitivity reactions. And in the case of penicillins, they don't actually get metabolized by P450, but they have intrinsic chemical reactive. That beta-lactam ring in the heart of the penicillin molecule, remember it's just a 4-membered ring, it's really strained, and it wants to react with a protein and open up and relieve that torsional strain within the ring. And so they have a high reactivity with proteins to form adducts that then can directly activate this sort of pathway of peptide presentation, T cell activation. Okay, so most of the time drugs acquire that reactivity by metabolism, but then there are also certain drugs that possess intrinsic chemical reactivity and activate this pathway. But the problem with that theory is that there are also drugs that don't form reactive metabolites and don't necessarily possess inherent chemical reactivity of a type possessed by penicillin. And so this led to the so-called pharmacological interaction hypothesis. And so this explains certain drugs such as sulfamethoxazole oxypurinol, which are known for causing some of these serious cutaneous adverse reactions. And what's going on here is that the parent drug seems to be binding to the peptide HLA complex in a non-covalent manner, and it either binds to the HLA complex or perhaps it also binds to the T cell receptor directly to activate the T cell directly. Okay, so this doesn't require metabolism, but for some drugs it's complicated because some drugs seem to have a direct pharmacological interaction pathway of action as well as a metabolite mediated response. So sometimes T cell activation can be quite complex. Now then there's a third theory that was sort of proposed really for abacavir, the antiretroviral drug found to produce this unpleasant hypersensitivity reactions, which is a little bit different to other types of T cell mediated adverse reactions. It doesn't necessarily fit into those three categories. But what's going on here is that the drug is thought to cause a change in the binding of self-peptides, so that in people with these inherited HLA variant forms, they're presenting different peptides to the T cell receptor. So it's activating T cell mediated responses in a way that doesn't normally occur in other people that lack that particular allele. Okay, so how does all this help us understand the clinical features of severe skin reactions? Well, there's the dominant view where there's this naive priming model where exposure to a drug results in the priming of T cell pathways, particularly in the lymph nodes, and then there's this migration of these primed T cells out to the skin, and then waiting for subsequent exposure after some period of time, and that's where we get this activation of those primed TRMs, the memory T cells, and we get this scar. But then there's also this heterologous immunity model where viral exposure in early life has a similar effect, and then subsequent exposure to a drug, even on the first exposure, can activate scar. These are pretty worrying conditions when you think that you may never have used a drug previously, but you can still have these serious conditions. Okay, so all of that is really just introduction to the pharmacogenomic aspect of this, and this is that people have found these various HLA risk alleles, and this is just a few of them. Have a look at this paper by Gibson if you want to know more about it, but there's a whole bunch of these. And they differ considerably in their prevalence in different populations. Some are dominant in Europeans, some are dominant in Africans, some are dominant in different Asian populations. And so the different prevalence of some of these variant alleles accounts for ethnic differences in susceptibility to some of these drug-induced hypersensitivity reactions. So this is basically the question: should we be genotyping people of different ethnic groups for some of these HLA variants before we prescribe some of these pharmaceuticals to them? Okay, that's just making the point that within Australia there is significant prevalence in some Asian populations for some of these HLA variants that have been associated with various drug-induced forms. But one of the reasons why there hasn't been as quick clinical uptake in this field as in other areas of pharmacogenomic testing is the fact that sometimes the genotype of an HLA allele is not that valuable in identifying at-risk patients. And so it's pretty good for predicting abacavir hypersensitivity syndrome, that HLA-B star 5701 allele predicts about 55% of adverse reactions to abacavir. But in terms of predicting vancomycin-induced eosinophilic and systemic toxicity, not so good. Only about 20% of cases are predicted by genotyping the HLA-A *3201. And then in the case of allopurinol-induced dermatological toxicity, that allele HLA-B *5801 predicts only 3% of cases. And the reason is that there's just a lot of factors contributing to an individual's risk for these conditions. So it's all sorts of factors, environmental factors, ecological factors, lifestyle factors that come in over the top of the genetic factors to influence your risk to some of these conditions. Okay, well I think I'm out of time, but that's the wrap up for pharmacogenomics. So hopefully these lectures have given you a bit of appreciation for how modern genetic testing can help us use drugs more safely in people and minimise unusual toxicities and adverse effects. Thanks very much for your attention. I look forward to seeing some of you this afternoon in the seminars. See you later. Bye-bye.
Use all these two theories because I think the drugs like here in these two theories they work the same way, like they all bind to.
No, it's changing it's changing the nature of the peptide that's presented. See that purple there? It's presenting a different peptide instead of the black one. Different peptide fragment is dominating the T cell receptor response. So
Oh, okay. So
it's changing a repertoire, which just means that different peptide fragments are being presented to the T cell receptor.
so this one requires the antigen presenting cell to process the drug and then present to the TCR, while this one doesn't need it.
Yeah, I don't know how that decision is made by a cell as to which peptides get presented to the T cell receptor, but with this hypothesis, the binding of the drug changes the peptide preference here.
Okay, I see.
This is my understanding anyway.
Yeah, it makes
Yeah.
sense.
Okay. Yeah. Thanks.
Thank you.
Yeah.